Swayam Prabha. Digital India. Educated India.
Welcome to lecture 3 of module 6 on TALEN technology. Today we are going to discuss about the applications of TALEN in diverse fields, particularly with respect to diverse organisms. So, before we move into the various specific examples of TALEN applications, so let us look into the promise and potential these technology holds overall. We know about TALEN as a Intel based engineered nucleus which can cause double strand breaks, and we can use the cell's internal repair methods triggered by these double strand breaks to insert DNA fragments as well as create point mutations. So, one of the potential or promise of TALEN obviously is creating mutations and genetic insertions, and TALEN have been employed to produce non-homologous and joining mediated mutations as well as homology directed repair based insertions in a range of species, generally with high efficiency while utilizing the double stranded donor templates. Somatic and gametic cell manipulation can also be carried out with TALENs. So, TALEN mediated gene DNA insertions into human somatic pluripotent stem cells as well as embryos and eggs are possible. The other promise or potential is in the area of creating knock-ins, knockouts as well as gene therapy. So, like zinc finger nucleases, TALEN has also been used widely in research to carry out genome editing in several species to produce disease models and knockout and knock-in animals in disease biology and to generate TALEN based genome editing therapeutics. And even beyond gene editing, we know TALEN can carry out tail based constructs can carry out many other functions. When fused with functional domains such as for base editing, then transposition, recombination, transcriptional activation or repression, etc. So, let us see the timeline for the first application of TALEN in laboratory animals. So, rat is the first animal in which TALEN was applied in August 2011 by Deschen et al. Then the next year it was used by Carlson et al. In 2012 in pigs directly, and mouse by Sang et al. In 2013. Then also rabbit by Song et al. The same year, and in 2014 Liu et al. Used these technology in monkeys. So, this is in brief the timeline for the first applications of TALEN in some of the standard laboratory animals. The first knockout of gene in rats was done by TALEN by Tesson et al. In 2011, as I already told you in the timeline discussion. So, Taschen and his colleagues first successfully created a gene knockout rat using these customized TAL effectors. They designed and assembled talents to exon 2 of rat IgM and tested their ability to alter the IgM locus in rat S16 cell lines. TALEN knockout rats lacked both mature B cells and secreted immunoglobulin. TALEN has been successfully used in production of both knockout and knock-ins in living cells and organisms by introducing tiny insertions and or deletions, indels into the genome. Non-homologous end joining frequently causes gene disruption, which we call as knockout. Alternatively, homologous recombination allows for the precise gene deletion or introduction in the targeted site, which is opted for TALEN based knock-in production. TALEN mediated knockout of zebrafish PRKDC gene. Zebrafish are model system for investigating human diseases such as cancer. Effective immunocompromised strains of zebrafish in tumor xenograft research is widely used. In mammalian cells, PRKDC gene encodes the DNA dependent protein kinase catalytic subunit. The catalytic subunit participates in DNA non-homologous end joining, without which it causes severe combined immunodeficiency or SCID. Zhang et al. Developed a PRKDC null or minus SCID zebrafish model for tumor xenotransplantation experiments. The zebrafish PRKDC genes exon 3 was selected to be targeted by two talents for this purpose, and this causes a frame shift mutation that rendered the gene completely inactive. After genome editing by TALEN, further analysis showed that the SCID zebrafish did not have functional T and B cells, which was consistent with the SCID mouse models observed abnormalities. So, this is in details the experiment for TALEN mediated knockout of zebrafish PRKDC gene, which has 86 exons and around 12,623 base pairs, and you can see here the different exons, and this is exon 3 to which this TALEN construct is targeted. And as you can see, we have TALEN F and TALEN R, which are designed to bind across the DNA strands so that the fork1 domains come opposite to one another and dimerize and facilitate the cleaving of the DNA. And here in B, these TALEN construct is delivered by microinjection of capped mRNA into yolk at one cell stage, and then you have these F0 founders which are crossed with the wild types, and the F1 founders are established as a result of these, and genotyping is done to confirm the successful crosses. So, step A you can see here is the generation of TALEN constructs. Exon 3 of the PRKDC gene was targeted with the 12 base pair spacer flanked by left and right TALEN arms. Mutagenesis by microinjection in step B of capped mRNA and strategy for the genotyping of the F1 progenies as already told to you. And in C you can see the sequence conformation of the mutations with 2 nucleotide deletions here and 6 nucleotide deletion followed by 5 nucleotide insertion. Both mutations cause a shift of the translational frame. So, here you can see the Western blot analysis for PRKDC Kidney and spleen were dissected from several geographies, and the pool samples were processed for the Western blotting experiment. And C, you have the PRKDC positive, and in P you have these PRKDC negative. These are not present over here. You can see here this is positive, but no any visible sign of PRKDC here in this Western blot analysis, and beta lectin is used as a control, positive control, which is so that we can know that the experiment is going on correctly. And these experiments successfully showed the effect of the talents. And here you have this is immunohistochemistry experiment for PRKDC, which was done with the kidney dissections, and the visual evidence supported the Western blot analysis as well. Let us also now look into other capabilities of talents and their applications, particularly knock-in red generation, where TALEN has been used. Leon et al. In 2014 reported the successful gene targeting knock-in in rat by TALEN mRNA injection in one cell embryos. TALEN was designed to target the PA4760 mutation in the rat glucocorticoid receptor or GR gene named as NR3C1. The mutation inhibits glucocorticoid receptor dimerization, namely GRDIM. So, a mutation in this gene will inhibit the dimerization of glucocorticoid receptor. Out of the design 18 TALEN constructs by in silico method, they selected 3 talents, TAL3, 6 and 13, and assembled them physically into heterodimeric FOC1 expressing vectors. So, TALEN design and evaluation of cutting efficiency in red glioma C6 cells. This is the schematic of these rat NC3R1 gene. So, you can zoom into this area of mutation of PA4760 in exon number 3. The first nucleotide of the 476 codon is highlighted in blue. The TALEN binding sites of the TAL3 are highlighted in green in both the left side and the right side. T-endo1 assay results, it showed that pooled DNA from C6 cells transfected with either right, left or right and left TALEN monomers, which are marked as R, L, was amplified and treated with T7 endo1 enzyme, and those with left right are marked as LR. Okay. So, you can see here 2 cut bands, 288 and 177 base pairs. You can see some of them visible over here, and this is the band for 465 base pairs, and which is being cleaved into 2 as shown in this figure. The M cells are mock transfected cells, and GFP are the GFP transfected cells, which are used as positive transfection control. Intensity of the cut bands are indicated for TAL3 and TAL6 pairs. So, here this is around 20% and this is around 16%. TAL3 transfected cells were screened. So, here PCR amplicons of the region around the PA76T mutations were subcloned into topo vector. Clones were isolated and analyzed individually. 4 point mutations and insertions are marked in red. So, you can see here 1, 2, 3, 4 and so on. Cells transfected with TAL3 presented mostly deletions ranging from 1 to 16 nucleotides. So, here you have 1 base pair minus 1 base pair here. Okay. So, this is the deletion, and you have say here minus 4 base pairs, and here you have larger ones minus 10, minus 13 and as big as 16 base pairs here. Donor molecule design and linearization. To generate glucocorticoid receptor dimerization GRDIM, knock in rats, a common donor plasmid was designed for talens 3 and 6 bearing the PA476 mutation along with 4 silent point mutations in each talen binding site to prevent further nuclease activity in the targeted alleles. The donor plasmid sequenced had also 500 base pair homology arms on 3' and 5' sites of the PA476T mutation to allow homology derived recombination. The donor carried 2 extra point mutations for rapid detection by enzyme digestion. An ALU1 site was removed and a high 3 site was added close to the P476T site. So, you can see here the wild type, and this is the TALEN tree left, and this is the TALEN tree right, and these are the sites we were speaking about, high tree, and this is TALEN 6 left, and this is TALEN 6 right. So, the upper DNA indicates the wild type sequence of the exon 3 of the rat NR3C1 GR. In blue, the residue A476, TAL3 and TAL6 binding sites are also indicated. DP is the donor plasmid sequence. Donor plasmid was synthesized with 500 base pair homology arms on both sides. The PA476T point mutation is highlighted in blue. Silent mutations of the DP are shown in red bold letters and are located in the overlap of TAL3 and TAL6 binding sites. High 3 site is highlighted in yellow color. TALEN mRNA injection in one cell stage embryo. TALEN mRNA, an excised linearized double stranded donor DNA containing point mutations and diagnostic restriction sites were co-injected into fertilized one cell stage embryos. Surviving embryos were implanted on the same day in the oviduct of pseudo pregnant females and allowed to develop to full term. So, this is the red genotyping. DNA from knock-in neonates was extracted by tail biopsy. Genotyping and sequencing was performed using primers specific to the targeted region. So, this is the wild type and donor plasmid DP. Then you have the DNA 1, 2 and 5 point mutations in the DP are indicated in red bold letters. The PA476T mutations is highlighted in blue. There are several other applications of TALEN for animal in animal editing. For example, there is a production of alpha 1,3 galactosyl transferase targeted pigs using transcription activator like effector nuclease mediated genome editing technology. This paper published by Kang group, and this is the discussion we had in the last module regarding the applications of zinc finger nucleases in generating HIV resistance cell lines. and you are all aware about of the importance of CD4 as well as CCR5 co-receptor, and the HIV virus can proliferate or enter a cell only when both of them are functional. If CD4 is functional but there is a mutation in CCR5, most famously known as the CCR5 delta 32 mutation, the structure of the CCR5 receptor is changed. The exposed transmembrane domains are absent in such kind of mutations, and due to these the virus cannot enter the cells, and thereby these kind of cell populations will become resistant to HIV. And we know that individuals homozygous for CCR5 delta 32 polymorphism show total protections against HIV type 1 infection, and those who are heterozygous will show delayed progression of HIV. So, once again let us have a brief recap of the CCR5 gene in HIV pathogenesis. This is a chemokine receptor which plays an essential part during HIV infection, and it acts as the co-receptor of the R5 trophic strains that usually mediate initial HIV infection. And in the absence of CCR5 on T helper cells, R5 HIV is unable to bind and thus cannot infect T lymphocytes. Approximately 10% of Caucasians are heterozygous and 1% homozygous for a deletion within these particular gene named CCR5 delta 32. In 2007, an American patient with HIV infection and acute myeloid leukemia AML was given bone marrow transplantation from a CCR5 delta 32 donor for AML therapy, leukemia therapy. And this was a kind of a accidental finding. Due to this bone marrow transplantation from a CCR5 delta 32 donor, the patient, which was HIV positive, got cured of his HIV infection. So, this is a accidental finding, and this has given us a clue about genome editing to modify the CCR5 receptor and thereby use this methodology as a HIV therapeutics approach. Mock et al. In 2015 designed a codon optimized TALEN targeting the functionally relevant first intracellular loop of the CCR5 receptor. This is designated as CCR5 TALEN. CCR5 TALEN mRNA was sent into the T cells lines PM1 through electroporation. Both TALEN arms recognize the 19 base pair target sequences within the CCR5 gene corresponding to the very short first intracellular hydrophilic loop of CCR5, a region expected to be sensitive for amino acid deletions or substitutions. CCR5 TALEN mediates CCR5 gene knockout at very high rates in primary T lymphocytes. The gene edited T lymphocytes are efficiently protected from infection with CCR5 tropic lentiviral vectors and also form fully replicating HIV-1 as reported by Mock et al. In 2015. So, let us look into the construct of CCR5 TALEN design. So, there is a left TALEN arm CCR5 L and a right TALEN arm CCR5 R, and here you can see the various transmembrane domains, total 7 in number of the CCR5, and you have these EL1, EL2 and EL3, and you can see here in EL2 the delta 32 mutation which occurs over there, and This is a semantic representation of the tail repeat variables die residues used for recognition of the CCR5 locus in A, and B is the schematic representation of CCR5 conformation and the region targeted by CCR5 TALEN, which is shown as red here. TM is transmembrane region, EL is extracellular loop, and IM is ICL is intracellular loop. TALEN binding sites in the genomic on target CCR5 and possible off target CCR loci is shown in figure number C. So, following these, they characterize CCR5 TALEN in the SAV susceptible T cell line PM1. So, you can see the percent non-homologous end joining going up post day electroporation, day 0 nothing, day 1 you have around 30%, then slowly day 2 you have around roughly 38%, but then it jumped to over 80% in day 3 and day 4 and little bit higher in towards day 7. So, non-homologous end joining induced mutations at the CCR5 locus over time can be seen in this figure A. PM1 cells were electroporated with 5 microgram mRNA of each CCR5 TALEN arm and genomic DNA was harvested at the indicated time points. The NSEJ frequency was determined by next generation sequencing. And in B you can see the correlation between loss of CCR5 expression and accumulation of NSEJ induced mutations, which is dependent on the amounts of electroporated CCR5 TALEN mRNA. CCR5 expression was determined by flow cytometry, NSEJ frequency was determined by NSEJ. So, as the positivity of CCR5 goes down, you can see the percentage of non-homologous end joining is going up. Let us now discuss about tail based tools and their applications in diverse fields. So, you can see here they are being used to make recombinases, transposes, then activation and repression, methylation as histone modifiers, base editors and well known function of nucleases. So, we will discuss some of these. So, using functional domains from other proteins as fusion partners, various tail based tools can be generated. Apart from using nuclease for genome editing, these tools can be used in a targeted and sequence specific manner to modify the transcriptome, modify the epigenome, modify the genome and visualize repetitive DNA sequences like centromeres or telomeres. Mitotalene. Talene being totally a protein molecule, they can locate to and function in every cell compartment which is accessible for proteins. Therefore, talens have been used to perform gene editing in mitochondrial DNA of human, mouse and plant cells. A mitochondrial targeting signal MTS is fused to the N terminus of talene to allow localization inside mitochondria. In mitochondria, pathways NSEJ and HDR for DSB repair are not functional. Therefore, DSB lead to rapid degradation of mitochondrial DNA and loss of the mitochondria. Intramolecular recombination may aid in DSB repair. However, this results in partial deletions of the mitochondrial chromosomes. Mitotalene can either be used to specifically deplete one type of mitochondria in a heteroplasmic cell and thereby increase the percentage of healthy mitochondria, or they can cause recombination events that results in deletions in the mitochondrial chromosomes. Talene offers advantages of delivery of mitotalene into mitochondria, which is not so efficient with CRISPR based system. as it requires a co-delivery of the protein and the RNA components into the organelle. Presence of defective mitochondria, mutant mitochondria at 60 to 90% in a cell causes a wide range of different diseases in the organisms. The treatment of cells with TALEN reduce the percentage of such defective mitochondria to 40% or less, in many cases even below 20%, thus bringing them well below threshold for a pathological phenotype. So, these are some of the tail based editors. The antibacterial toxin DAAA, which stands for double stranded DNA deaminase toxin A, acts as a cytidine deaminase and promotes CG to TA conversions with a high bias for 5' TC context. In contrast to other known cytidine deaminases, double stranded DNA deaminase toxin can act on double stranded DNA molecules as substrate. Triple DA has been fused with tails. Okay, so DDA is here and it has been fused with tails to exploit the function of base editing. A tail base editor is composed of a tail repeat region, the N and C terminal regions from a tail both truncated to 146 and 40 amino acids respectively. One half of the cytidine deaminase domain of triple DAN or C terminus and an uracil glycosylase inhibitor UGI. So, this is the construct basically, part of UGI, part of triple DA and these tail construct. Fusing an uracil glycosylase inhibitor to each tail base editor further increased the overall efficiency substantially. Now, there are issues of toxicity associated with this entire approach. So, those has to be tackled. The fusion of the deaminase domain of triple DA and the DNA binding domain of tail was found to be toxic to cells, as the deaminase targeted multiple chromosomal sites during the search process or when released from the tail after degradation. The deaminase domain was split into two non-toxic halves, triple DAN and triple DAC, and fused them to form two oppositely binding tails. This restricted the protein activity to the targeted region where both halves can assemble into a functional unit, which ultimately reduced the toxicity of the tail triple DA fusion protein. The recommended breaking points being either between amino acids portions 1333 and 1334 or 1397 and 1398, named G1 333, G1 397 respectively. Since the editing range of the tail base editor pair is governed considerably by this split point, it is recommended to either evaluate which one of the two might be best suited for the planned experiment or to use both. Use of mitochondrial targeting signal can deliver the tail base editor inside mitochondria as well and carry out base editing there. The optimal spacer length between the two tail binding site is indicated in green. You can see here around 14 to 18 base pairs. The approximate editing range for a tail base editor pair utilizing either the triple DA halves G1333N or C N and C or G1397N and C on the target sequence with 14 base pair spaces are shown in orange. This is the editing range and this is G1333 and this is G1397. Tail base editors facilitate CG to TA conversions and have a strong preference for 5' TC contexts. So, with this we come to end of this part. We will continue this lecture in the next part. Thank you.